So welcome to everybody this afternoon. It's my real pleasure to introduce today's speaker, Professor Harvey Lodish, who is a guest of the Mortimer and Raymond Sackler Institute of Advanced Sciences and Studies, And it's actually the second time that Harvey, I'm going to call you from now on Harvey and not Professor Lodish. So it's the second time that Harvey is visiting Israel. And actually the first time that Harvey visited Israel was 20 years ago, when also then he was a guest of the Mortimer and Raymond Sackler Institute of Advanced Sciences Studies. So it's the second time that Harvey's been to Israel, both in the same capacity, and we're really, really happy that you returned to visit us here. So Professor Harvey Lodish is a member of the Whitehead Institute of Biomedical Research. He's a professor of biology and professor of biological engineering at the MIT in Boston. Harvey is a leader in the field of molecular and cell biology. He has identified and characterized numerous surface membrane proteins that play a role in blood development, signaling, glucose transport, lipid metabolism, and more. And here are some things that are not officially written in Harvey's CV, but I learned them yesterday. So already at a very young age, Harvey showed extraordinary qualities. He was the top student in his Hebrew class and in the high school class, and he was marked to be a top rabbi. However, he then decided not to go along with the rabbinate plan, but rather pursue a career in science. So this is maybe probably a great loss for the rabbinate movement and community, but definitely a huge benefit for the scientific community. So Harvey received his BA degree with highest honors in chemistry and mathematics from Kenyon College in 1962, and his PhD degree in genetics with Norton Zinder from the Rockefeller University in 1966. Following 2 years of postdoc research fellowship at the MRC Laboratory of Molecular Biology with Sidney Brenner and Francis Crick, he joined the faculty of the MIT Department of Biology. He's a founding member of the Whitehead Institute. Harvey joined the MIT faculty in 1968 and has been a professor of biology since 1976 and a professor of biological engineering since 1999. He is the lead author of the widely used textbook Molecular Cell Biology. As we all know, we read it and we teach it and our students learn it. The last edition appeared in April 2012, and I hear that there is a new edition about to come out soon. This textbook has been translated into 10 languages. Harvey is a member of the National Academy of Science. He's a fellow of the American Association for the Advancement of Science, the American Academy of Arts and Science, the American Academy of Microbiology, and associate foreign member of the European Molecular Biology Organization. Harvey is chair of the Scientific Advisory Board of the Massachusetts Life Science Center, the group charged with the oversight of the state's 10-year, $1 billion investment in life sciences. And Harvey was also a founder and scientific advisory board member of several companies, Genzyme, Aris, Millennium, and Allozyme. Now, on the personal note, the mission of a great scientist, whom Harvey is really twofold. The first is to advance your own research, your own laboratory, publish papers, write theories, come up with theories, and alike. And the second mission is to generate a line of young independent investigators, successors, that will advance science beyond what one individual can actually do. So regarding the first aspects, there's no question. Harvey certainly generated first-class science. He published more than 500 papers in 600? Really? Oh, thank you. I mean, I looked in the PubMed, so some are referenced differently, but so even more. Oh, I'm sorry. All right. Right, and in the second aspect, not less important and probably more, Harvey generated the ultimate soil for the growth of a future generation of scientists with top publication and leading positions. Over 150, so maybe this also changed since this was written, but 180 MD, PhD, and MD students and fellows have trained in his laboratory, including myself, and Gerardo, who's here, and Yoav Hennis, who will probably come soon, and Aaron Chechanover, who comes along the second category that I'm going to mention. And two of those fellows, Jim Rothman and Aaron Chechanover, received both Nobel Prize and Lasker Awards. So that's a big pride. That's a very big pride. And we met with Aaron Chechanover yesterday, who told us and Harvey that the period in Harvey's lab was really the best scientific period he had ever encountered, with the freedom he got from Harvey, the infrastructure, the support, the wisdom, everything. I'm speaking now for Aaron Chechanover. And four of his students, so Rothman, Chechanover, David Hausman, and Charles Zucker, have been elected to the National U.S. Academy of Sciences, and others, including Jeff Lyer and Diane Rith, to Institutes of Medicine. So the other unique feature of Harvey is the very, very good relationships that he has maintained with his former students all along the years and trainees along this international pathway. So Harvey, we're really indebted to you, and we have so much more to learn from you. And thank you so much for coming here and being with us.
Thank you. Okay.
It's yours.
Well, thank you so much. I mean, there really is one other, accomplishment which I have to mention. There's been close to 60 women that I've trained in my lab, of whom 26 had small children when they were working in my group. And I can tell you they've all gone on to successful careers, including Dorit. So anyway, it's great to be here. Thank you. I don't deserve these introductions, but no. Anyway, I'm going to talk to you about a project that has been going on in my lab for about 7 years, which started with a medical problem, trying to understand how corticosteroids work in this bone marrow failure disorder that I'll come to. And what it's led us to is a fundamental question in developmental biology and in cell biology, which is cell division and its role in differentiation. That is, when a cell divides, a stem cell or a progenitor cell, how does it know what identity to take on? Does it take on the identity of the parental cell, in which case it undergoes self-renewal, or does it take on the identity of a cell that is more further along the differentiation pathway? And that, as I'll explain, occurs in many developmental pathways. It's not well understood. We're beginning to understand that in the context of a red cell progenitor, and in the process developing or actually finding old drugs and repurposing them that can and will be used in the clinic to treat these rare bone marrow failure disorders. So it's the kind of problem that I love to work on, because it's really a detective story, and one in which very few people had thought to work on. Now,
Use the mouse that we...
Use the mouse. I was going to say I am genetically short. I mean, I can't help it. I come from a family of short people. But basically what we're going to be talking about is hematopoiesis, the process by which a hematopoietic stem cell gives rise to multiple differentiated progeny. And we're going to focus particularly on the red cell pathway. And if we start with the stem cell, it's capable, it's considered to be an immortal cell. And what we really mean by that is not that the cell lives for the lifetime of an individual, but when the cell divides, on average, at least one of its offspring retain stem cell ability. So that the number of hematopoietic stem cells in adult remains constant throughout one's lifetime, or may decline with age. In early development, the probability of self-renewal is higher because the body is making more stem cells to populate the bone marrow. But what we're going to come to are these various intermediate cells, which, as you'll see, have limited self-renewal capability. And this figure, and I forget which paper I took it from, illustrates the hierarchy of self-renewal. I believe this reflects the intestinal stem cell pathway, where you have in the crypts cells that undergo self-renewal and that at least one of the daughters is a stem cell. But you get these rapidly dividing, transient amplifying cells that have limited self-renewal capability. And eventually they form cells with no self-renewal capability that go on and form differentiated cells. And it's embedded in here is the solutions to a number of diseases. So when we focus on red cell development, again, we start with the stem cell, and we see two types of progenitor cells. The later cells, which I'll describe first, the so-called colony-forming unit cells, are totally dependent on the hormone EPO. And we can recognize these cells by their ability to form erythroid colonies in methylcellulose. That is, if you plate a cell like this, and we have purified them, you add EPO and wait 3 days, you get a colony of 30 to 50 hemoglobinized cells. And this textbook picture, you know which textbook it's from, shows what goes on in this cell population. That is, the cells have on their surface EPO receptors. If they see EPO, they go through a fairly stereotypic process of 4 to 5 cell divisions. Midway in it, they turn on hemoglobin. They eventually lose their nucleus and form mature erythrocytes. In the absence of EPO, they undergo apoptosis. And we have been studying this process for 30-some years, ever since we cloned, or Alan DeAndrea in my lab cloned the EPO receptor many years ago. And EPO, for those of you who this is Lance Armstrong. This is one of the drugs he abused. So don't try it yourself. But let me go back to this slide. The progenitor that's going to occupy most of our discussion is this earlier progenitor, which is a kind of transient amplifying cell. And as you can see from this arrow, it can undergo limited self-renewal and also acquire the ability to generate daughter cells that are the CFUE cells. And it's precisely this decision that we're going to be discussing initially in the context of this bone marrow failure disorder. And EPO, as I should have mentioned, is used to treat a number of anemias. At one point, it was the largest selling drug in the world. It earned Amgen close to $9 billion a year. Do you know how much $9 billion is? The gross national product of Israel is $300 billion. Okay? So the worldwide sales of EPO equal 3% of Israel's total goods and output. That's quite remarkable for a protein. But that is used in kidney failure, where the kidney fails to produce enough EPO, in many cancers where the patients are anemic for one reason or another. But many anemias do not respond to EPO. And that's because there are not enough progenitors to respond to all the EPO even that the body makes to make enough red cells. And the one that started me on this journey is a genetic bone marrow failure disease termed diamond blackfan anemia. It's a ribosome haploinsufficiency disease. That is, it's caused by having one inactivated ribosomal protein gene, can be RPS19, it can be any of several large or small ribosomal protein genes. And it's a pure red cell aplasia. The babies develop normally in utero. Fetal erythropoiesis is normal. But when they're born, adult erythropoiesis simply doesn't turn on. And they have reduced numbers of both the early or the late CFUEs and the early BFUEs in their bone marrow. And this has been known, it's been a very difficult disease to treat because, as you can see here, many of these progenitors are undergoing apoptosis. So even though the body is responding by producing huge amounts of EPO, there simply aren't enough of these progenitors to go around. So the babies are anemic and they frequently die. And in fact, as part of what I do now, I talk to parents of patients either with the children with the disease or children who have died from the disease. So it's a very immediate issue. And where we started was this. That is, a clinician about 30 years ago thought that the disease had something to do with autoimmunity and treated it with cortisol or prednisone, a corticosteroid. And it works, but with enormous side effects. There are growth defects of all sorts, developmental defects. It does stimulate red cell production, but the patients don't do very well on it. Most of them have to be taken off. They're treated with transfusions and various other things, but as I said, many of them die. So about 7 years ago now, I was invited to a DBA meeting and gave a talk on how red cells were made, which is something I know about, and then asked people, well, how do corticosteroids work? I mean, here's a disease which at least shows some response to them. And the answer was nobody knows. So this is the kind of thing I like to work on. And I recruited, as I'll describe, Johan Flygerer, who was an MD PhD just finishing his thesis in Lund, Sweden. And I said, come to my lab and let's figure it out. And he was intrigued by it because in various experiments done in the 80s and 90s, it was clear that corticosteroids would stimulate red cell production, but again, no one quite knew how, and there was a lot of controversy as to how this happened. So I said, well, let's find out. And then we can understand both, well, we'll try to figure out how it works, and then see if we can find other drugs that might mimic it, and that will allow lower levels of steroids to work. So this is what I'm going to talk to you about. The first two topics have been published. The third is about to come out in Nature, and I'll spend more time with it. But basically what I'm going to tell you is that the corticosteroids work by triggering self-renewal of the BFUE cells. So over time, you make more of them. And then over extended periods of time, each BFUE will go on and make more of the later CFUE progenitors, which in turn will make more red cells. And then, as part of this study, we landed on hypoxia to do the same thing, HIF-1 alpha, which begins to make a certain amount of sense because cortisol is a stress hormone, and hypoxia is local stress. So what we believe is that this response mimics a normal cellular response to enormous stress, and I'll come to that. Then we decided to understand how it works, and and uncovered one of the genes that is upregulated by corticosteroids and by hypoxia, which is an RNA binding protein called ZFP36L2, which I'll call ZFP, which binds to and degrades many messenger RNAs that would normally be upregulated at the next differentiation step. And by degrading those messenger RNAs, it helps shift the cell towards self-renewal by blocking differentiation. And finally, and most recently, we screened for other molecules that might mimic corticosteroids, came up with drugs that activate the PPAR alpha nuclear receptor, which had never been implicated in red cells at all, or hematopoiesis for that matter, known as a lipid-lowering agent. But these are the ones we're taking into the clinic, and we know how they work. Okay? So that's where we're going. If you fall asleep and wake up, you can get oriented. So, self-renewal. This is what we're talking about, this process here. So basically what Johann did is first purified CFUE cells and BFUE cells. These are mouse. We'll come to humans in a moment because we have it all in the human system. Basically using FACS, he could get cells which by colony-forming assays were 80 to 90% pure. And this was the key experiment that took us a while to understand. If you put the CFUEs in culture and add EPO, over 3 days they generate 30 daughter cells, and that's the 5 divisions they undergo. And contrary to what a lot of people thought, corticosteroids have no effect on these whatsoever. If you put the BFUEs in culture, again, it took us a while to understand the curve. Whether or not we added corticosteroids, they divide 4 or 5 times at the same rate. So the rate of division is not affected. In the absence of corticosteroids, the cells stop dividing around day 5, and each BFUE produces about 600 red cells. And I'll show you later the data, but basically what we determined is over the first 3 days, each BFUE produced after divisions about 20 CFUEs. And then over the subsequent 3 days, each of those CFUEs produced 30 red cells, roughly speaking. Okay? So very little self-renewal of the BFUEs. They go straight to dividing, differentiating, and so forth. In the presence of corticosteroids, the cells kept dividing. Eventually, they made, 40 times more red cells, but it took them 3 or 4 days longer. So just to illustrate this in a non-mathematical sense, I can show you differential equations. Okay? I can do that, I'm happy to, but you don't want to see them. Okay? They're linear differential equations, they're not partial differential. Never mind. But basically, if you start with what happens in the absence of steroids, you start with a BFUE. It may undergo 1 self-renewal division, maybe 2, but eventually it gives rise to CFUEs, but there's no more BFUEs. So the CFUEs then go through for the next 3 days and make the differentiated red cells. So this is going from 1 to 600. In the presence of corticosteroids, the probability that each division is a self-renewal division increases. So here you see you go from 1, 1 to 2, maybe 3, maybe 4 BFUEs. Obviously it's actually 30 or 40 as we'll see. That over time make more CFUEs, that over the next 4 or 5 days will make more red cells. And that is what explains this curve. Okay? Do you all get this? This is the key concept. That is, when you self-renew, you will eventually make more red cells, but it takes longer because the progenitors stay immature for a couple of days longer. Okay? So from there we were able to move quickly. And And one of the things he did was to do high-throughput RNA sequencing and look for genes that were upregulated by corticosteroids. And many of them didn't make too much sense. Although here's one, c-KIT, that did make sense, because we know KIT is involved, it's the receptor for stem cell factor, it's a protein tyrosine kinase, it's essential for self-renewal. So it was interesting that it would be upregulated. Similarly, BCL2, the anti-apoptotic protein. But we did a lot of computational analysis. We did not find in the promoters of these genes binding sites for corticosteroid receptors. And that was not surprising because they tend to bind to enhancer regions. But what we did find are potential binding sites for HIF-1 alpha, the oxygen-sensing transcription factor. And that became very interesting because that also is involved in self-renewal. And basically, to understand this oxygen-sensing transcription factor, it's made at high levels in BFUEs. At ambient oxygen, it undergoes oxygenation of a particular proline, which triggers its degradation. But in the presence of inhibitors of prolyl hydroxylase, this degradation is blocked, and the protein survives, moves into the nucleus, and turns on genes. So it's a transcription factor that is sensitive to anoxia, which is again a local form of stress. And these inhibitors are chemically available. The one prolyl hydroxylase inhibitors, the one we used in these initial studies is a pan-prolyl hydroxylase inhibitor. And as you can see from this curve, it exaggerates what happens just in the presence of corticosteroids. You have to have the steroid there, but if you have the steroid and the prolyl hydroxylase inhibitor, you get another 4 or 5 divisions and a further increase in red cell production. Now, I can show you this curve again, but this is a heroic experiment. One of the most elaborate experiments that I've ever seen done. Because in each day of culture, he took the cells and plated them for their progenitors. And I'm going to do it here, okay? If you lump together small BFUEs and large BFUEs, we start with 100 purified BFUEs. This is to show you that I'm not lying, okay? So we start with 100 purified BFUEs, of which by colony assay, about, 60, almost 70% are colony-forming BFUEs. So roughly 100. In cultures with nothing, this number may get up to 200, a little bit of self-renewal, but then they die off. If you add corticosteroids, this 100 eventually becomes 800 and 1,000. So you can see the number of self-renewal divisions, on average 3. If you do the same experiment, but add the prolyl hydroxylase inhibitor, the number of these BFUEs rises to 2,000 or 4,000 at later times. So you can see the enhanced self-renewal, and this is reflected in the peak numbers of the CFUE offspring. The 100 BFUEs in control cultures gives rise to 1,000 progenitors. In the presence of corticosteroids, you get 28,000, and in the presence of both drugs, you get 200,000. So when I talk about self-renewal and show you a cartoon like this, I'm serious. Okay? So it works. And what makes it encouraging is there are now 2 clinically tested prolyl hydroxylase inhibitors that are highly selective for prolyl hydroxylase 2, which is the one that regulates HIF-1 alpha. They were developed by these two companies as EPO substitutes. And the only reason we're not taking them into the clinic is they haven't been tested on kids. But there's every reason for thinking they will work. So that's pretty much the first part. I just want to finish with the fact that we can do the same thing with human cells. We developed, and it's coming out in a few weeks as part of the later story, we now can take mobilized blood CD34 positive stem cells, hematopoietic stem cells. These are the cells that you would use in a bone marrow transplant. We can put them in a 21-day culture. We get over, well, we're now up to 100,000-fold expansion, and we make pure red cells. Okay? I'll just show you some pictures. About half the cells lose their nuclei. They're pretty normal-looking red cells. They have the size of mature red cells. They are fully hemoglobinized. They have the amount of hemoglobin that a mature red cell would have. They express the right globin chains, hemoglobin alpha, relatively little fetal hemoglobin, very similar to normal red cells. And we put them in immune-compromised mice, and they live as long as the human red cells live. Because mice have smaller capillaries than humans, human red cells don't live as long in a mouse as they do in a human, which is 4 months, but these cells live as long as human red cells. So we actually have them made in culture and they work. The details will come out. And just as an example of what we can do with this system, these are the 21-day cultures. If you add just dexamethasone alone, you get a certain level of red cells. But if you couple it with either the pan-prolyl hydroxylase inhibitor, I'm sorry, here, 100 micromolar of the pan, or 0.1 nanomolar of the clinical prolyl hydroxylase inhibitor, we get another 20-fold stimulation. I guess in this case it's about a 8-fold stimulation. So the point is it works. And that's the first part. So we understand how corticosteroids work in general. And then the next question is, in a molecular sense, how does it work? That is, what are the targets of the corticosteroids? Well, some of them I already mentioned. One is c-KIT, which is a surface receptor that we've known from genetics for many years is essential for BFUE self-renewal, or at least BFUE function. And Lingbo Zhang, a graduate student in the lab, decided to study this protein, this RNA binding protein, because it was the most heavily upregulated by corticosteroids. And what he started with, and we do a lot of this kind of computation, that many of the same genes were upregulated by corticosteroids and by prolyl hydroxylase inhibitors in these BFUE cells. So these immediately attracted attention because these hypothetically could be genes having something to do with self-renewal. And the one he focused on was ZFP, because these are RPKM values, which is a measure of messenger RNA abundance. So in control cells, there's 100 units, and it goes up to 400 or 300. And we used a whole variety of cortisol analogs just to eliminate a lot of artifacts. But basically, this is the one that he landed on. And as I'll tell you, and I'll go through this briefly, it binds, it's upregulated by the corticosteroid receptor. So the corticosteroid receptor binds to enhancer segments of this gene. If you knock the gene down, you get differentiation, but you do not get corticosteroid self-renewal. So it is necessary for self-renewal induced by steroids. It is not sufficient because if you simply overexpress it, it does not in itself stimulate self-renewal. But it binds to, and I'll show you negatively regulates, a number of genes that would otherwise promote differentiation. So again, the way to think about this is analog to digital. That is, these BFUEs are getting a lot of signals, both from outside and from inside, and they have to integrate this into a single decision. That is, do my daughters become like me, or do they differentiate? And this is part of the signal. It's by no means all of the signal. So basically, in terms of the basics, this is a protein that's high in these BFUEs and goes down with differentiation. It goes down, this is normal differentiation. In culture, it goes down if you don't add corticosteroids, but in the presence of corticosteroids, it's maintained at a normal level. So that is nice. It says at least it's consistent with corticosteroid-induced upregulation. We know members of this family bind to AU-rich regions in the 3' untranslated regions of messages and trigger their degradation either 3' to 5' or 5' to 3' or both.